Welcome, everybody, to the February 18th, 2025 edition of Taxonomy Tuesdays, sponsored by the New Jersey Mycological Association. I'm your host for this evening, Mike Inskeep, and I have on my lap my helpful assistant, Oksana, who's keeping my lap warm. So, I'm interested in knowing who has observations they'd like to share this week. I did get shares. Hunter and Dave both alerted me that they have things they want to share. Do other people have things they'd like to share?
Mike, I just wanted to interject here. There's a NAMA presentation that starts at 8 p.m. I think I'm going to want to attend it.
Okay. All right. So is there anyone else who has observations they'd like to share?
Dave, you know that like all those are recorded and they're putting them up pretty quickly to be seen.
Oh, you're right.
Yeah, I mean,
Yeah. Honestly, I don't think that the thing that's being presented tonight is something where I'm going to need to ask a lot of questions. But I wouldn't mind attending it. Although, well, I'll make my decision at 7:55, I guess.
Okay, cool. All right. So why don't we get started then? If other people discover observations they'd like to share, let me know. And maybe I'll start with Hunter. Does that sound good, Hunter?
unless Dave wants to go first because of the meeting, but usually he doesn't. But do you want to go first, Dave?
I can go first. Yeah,
Okay.
there's only
cool.
really one mushroom to talk about. It's not
All
anything recent, but is Brandon here, by the way?
He is.
Oh, good. Yeah, this is something we've discussed with
All
Brandon.
right, so let me get it up here and I'll share my screen. So I'm going to start with your observation, Dave.
Right. Now, this is a species that I think is soon going to be given a definite species name by Alfredo Justo. It's a species of Pluteus, and he's calling it now Amphibious group. Now, the interesting thing about this mushroom is, aside from it being, representative of what will be a newly minted species name, is that it's placed in section Celluloderma. Now, the three sections of Pluteus are distinguished microscopically. Section Pluteus has metuloids, which are pleurocystidia with thick walls. The other two sections, Hispidoderma and Celluloderma, do not have metuloids. They have pleurocystidia, but they're not thick-walled, and they're usually not ornamented either, with like things on the apices. But a major distinguishing feature for section Celluloderma is supposed to be the pileopellis is composed of globose to subglobose to polygonal elements. Now, this thing, Amphibious group, is put into section Celluloderma. And I think I have here one. If you keep going through these photos, I think the next one, or eventually you're going to see that the pileopellis. It's not that. I think that's a. That's probably a pleurocystidium there. It looks like a bowling ball, right? But it's thin walled. So it's not section. This is not anything in section Pluteus. There's another thin walled bowling ball sort of thing. And these are probably Chylocystidia here. But now we get to the interesting thing. The pileopellis for section Celluloderma is supposed to be these globose to subglobose to polygonal elements that form like a mosaic sort of. But look at these things. They look almost like some sort of cystidia. There are these elongate subfusiform, I guess you can say, elements that are this doesn't fit in at all. So I actually sent an email to Alfredo Justo a few days ago, and he responded. He's a very nice guy. He's very helpful. He's one of the professionals out there who's not a black hole. You can engage with him, and he will engage with you. Section Celluloderma includes a few sort of atypical species. One of them is, some of them are in the group Ephebius group, and then there's another group called the Hispidus group. And these mushrooms have a paleopellis that is not typical of Celluloderma. They have these elongated elements. Now, so you might wonder, well, why isn't this put into Hispidoderma, which has a cutis, which is elongated elements. But Hispidoderma, the elements are perpendicular to the cap surface. Now, my section here was not good enough to really see how these things are aligned with respect to the rest of the cap surface. But anyway,
Hey, Dave,
yeah.
Can I ask a question real quick? So are these potentially Pileocystidia?
No, I put that in a comment on the next observation that we'll see, which is from Django Grootmeyers, who's recently completed a PhD in mycology. This looks like Django's here.
Yes.
And this is the same group, the Ephebius group. His pictures are a little better. He probably did a little bit better section. So Django thought, though, that these elements were erect, perpendicular to the cap surface. And apparently they weren't. It's just the way that they got mounted. They looked that way. But this is his observation is also a Ephebius group. So there's been a few attempts over the years to make a separate section. Elsa Vilenga proposed a new section for these things that would otherwise be put into Celluloderma, section Celluloderma, but have the wrong sort of paleopellis. And then there was also Singer apparently proposed a subsection of Celluloderma. So then one wonders, why not? Why not make a separate taxonomic category for these sort of strange species strange because of the microscopy of the paleopellis does not match up with where they would be otherwise placed. And so anyway, the answer is this. They are not Hispidus group and Ephebius group are not closely enough related genetically in order to form any sort of a reasonable clade. So they remain in section Celluloderma, even though the microscopy runs contrary to one of what is supposedly the defining characters of the section. So finally, I'm getting a little bit of closure on this.
And Dave, where was this growing? Was this terrestrial?
Well, this was Django's, I believe. Mine were found on my own property, terrestrially, and certainly from buried woody debris of some sort. But yeah, I've been finding mine in the same spot. By the way, this one here was not sequenced, but it came from the same square meter, where at least one other collection that looked exactly pretty much the same, was sequenced, and Fredo got involved in that discussion and said that it was Ephebius. In fact, it was sequenced by Fredo. I sent him the material, and yeah. So there's Fredo saying that this is so one of the you can sort of get a handle on these morphologically, maybe, because See the way the cap cracks? That's not true striations. Those are. This is just rimos. Rimos. You can say here these little radial lines that develop. They're actually cracks. Now another thing that's strange is that this is actually a trait that's often associated with section. What is it? Hispidulus. What I put it into the email. But anyway, one of the other sections. So, any confusion out there that you might run into between section celluloderma and hispiduloderma, I guess it is. I forget. Is understandable.
Hispidoderma, I think it is.
Yes.
Hispidoderma. Thank you. Yes, it's to be expected. Because these classifications are not morphologically well defined. So it's really a product of genetics that this thing is in celluloderma.
That's cool.
Yeah, I thought that was kind of interesting. I thought you'd enjoy that one.
Yeah.
Was that
Sorry, Chris, real quick. Where did you find a tree of section hispidoderma?
You have to go on to the I don't think it's shareable. So it was a NAMA presentation that one of which is of the type that Hunter just alluded to. So NAMA will send you an email. If you're a NAMA member, you get an email of recent webinars, and one of them is the one from Alfredo, and he shows the tree in that webinar. I looked for it online. I couldn't find it. It's not published yet. So a lot of this that I'm talking about is not yet published, which is one reason why we don't have a name for this species yet.
You had a question, Christopher?
Oh, was the material you took off the Pileopellis, was that from dried material?
I forget. I don't think so. I have a hard time working with dry. I mean, the pros will tell you, "Oh, it's easier to work with dried material," but I guess
this is why I'm asking, because I've run into this a few times now recently, where everyone I only dry my stuff out. I'm like, I think that's such a pain in the ass.
Yeah. Well, for those of us who don't have like highly specialized instruments and lab technicians to take care of the stuff, it's probably easier to take a fresh mushroom of this type and just peel a little bit of the Pileopellis off and trim it down with a razor blade and squash it in some Congo red, and then hopefully at least you get something as good as this.
I was asking because it looked good.
It actually, this one actually looked pretty good. Yeah, I got kind of lucky here, I think.
I think that's the best, best practice for getting Pileopellis microscopy, as well as Callocystidia from the stipe. Someone was talking about it recently, and you want to do a scalp of it, right? So you want to do that, laterally, right, or longitudinally.
The question I think is, what's the best way to trim down your scalped piece of material? But yeah, I do that with the stipe also when I'm looking for Callocystidia. I just try to get a really, really thin section of the stipe surface and squash it, and then you look on the edges, for the Callocystidia. They're usually not that hard to find. I've had pretty good success finding Callocystidia. By the way, the difference between these elements here, in my experience, which is, you have to take this with a grain of salt, and when you scope the Pileopellis of of a fruit body representing a species in section Pluteus. These elements are really, they're very distinctive. You know, they're elongated and tapered, like fusiform sort of. But the Pileopellis that I have seen for mushrooms in section Pluteus, it's just this mass of hyphae, you know, that they're all plastered together, and you can see they're all more or less parallel. And it looks a little bit different than this. They don't like stick out really distinctively like these guys. But this is a work in progress for me also, understanding microscopy associated with various species of Pluteus.
Cool. Thank you, Dave.
Okay, good. Yeah, I'm glad I was able to get that in. Thanks.
Oh, so Hunter, you're up.
Okay. This is a lichen, I believe, in Arizona. And I was hoping we could look at it. Maybe Jason would know what it is. I haven't had a chance to look at what it might be at all. But I have a second picture that morphologically looks really similar, but is bright green. And so I'm wondering if they're maybe the same. But one has received more sun or something, or if they're different. But they're really, really pretty. And if you zoom in real close, you can see the small little, they look like little ascomycetes, but I don't think that's what they are. They're a lichen, something, a structure in lichens that I haven't learned yet.
Can you guys hear me?
Yes.
So the first one, if you go back to the orange one, that might be a species we have in New Jersey. It's what I call Caloplaca subsoluta, I think, subsolulata, something like that.
Okay.
I think Caloplaca might be an old genus. I think iNaturalist is using an updated name. And you can see how these are definitely different species.
Okay.
Probably even different genera. I mean, this is Caloplaca squamulea might be the new genus. But the yellow one is different. I mean, this one you can see how it's kind of foliose. You see around the edges, it's got, a thallus with lobes.
Okay.
The yellow one doesn't. The yellow one the thallus is basically just kind of areolate. It's just a mass of apothecia stuck together. You can have variations in color for lichens, but usually they're not this dramatic. It's going to be different pigments, different chemistries that are creating yellow and orange. So I think they're different, but I don't recognize the yellow one. I'm guessing it's probably something that's out in Arizona, but not New Jersey.
These were really quite vibrant and nice to see. Would you mind typing the names you said in the chat for me?
Yeah.
There's a genus of lichen that we have observed out in Montana in Glacier National Park, and the east side of the park where it's a little drier.
Yeah.
I think it started with X, and I'm going to look through my records now and see what I can find, just as a suggestion.
Xantho Mendoza.
Maybe I forget. It's been a while. It's been well over 10 years. Let me check.
Yeah, now that I think about it, Caloplaca is a crustose species, and this orange one looks foliose. So the color orange matches the Apothecia match, but yeah, I'm gonna guess that neither of these are species we have in New Jersey.
That they're not.
Correct. Yeah.
Okay.
Yeah.
Oh, well, I'm excited. I have 3 different species here. That's neat.
And then this is just a bunch of areolate apothecia, like you were talking about on the last yellow one, right, Jason?
Yeah, maybe. I mean, a lot of these look like Apothecias. Maybe some of these are young. You can definitely see the outer rim. A lot of these. So that'd be my guess. I don't know if we it, something like this. I'm guessing is distinct enough where the iNaturalist AI would give you a pretty reasonable ID, and then you could just look up the species description.
Okay. I'll
Yep.
put them up.
The orange things that we found in Montana, Xanthoria, is what Karen came up with for those.
And there's
I'll check
this.
that out. Yeah, so this is from Mississippi, northern Mississippi, a couple weeks back when I was there like a month ago. And I think Brandon has showed something similar. And I also did not have a chance to really look into what it was. Kind of hoping someone else would know. Because I do think we've seen it here before.
You know what it was grown on by any chance?
If we back out, we might be able to tell. I think the whole log is. It was deciduous woods. I'm pretty sure only. Primarily. But
How big are like are these little clumps pretty small or?
no, I think this is a pretty decent sized log. Yeah, if I remember correctly, it's even probably 4 to 6 inches in diameter.
Yeah, something, hypoxylon ACA.
Okay.
If they're big globs, then maybe Kretschmer Maria, Duesta. I always mix it up. You need microscopy for these things.
Yeah, that's not a bad call on that one. I can never remember the genus on that.
Yeah, and I know that I'm familiar with that. I'll look into that
If
too.
you bring home fruit bodies of that species, you should, and you have trees on your property or nearby, you should dispose of the material properly, wrap it up in a little plastic bag or something, and put it in the trash. If the spores germinate near any of your hardwood trees, it's a parasitic fungus. It can do a lot of damage.
Okay, I did not bring it home, but I'll keep that in mind.
Yeah, and I'm trying to think of something a better ID because at that size, um, your Jack Rogerzellas, your annular hypoxylons, your even hypoxylon proper, the genus, it looks bigger than that. If that log's 4 to 6 inches, but could be wrong. There's species that I'm not aware of in those genera.
Oh, you think this is smaller than that? Is that or bigger?
Well, no, if you're saying it's 4 to 6, it's a matter of scale, right? You know, I don't know.
Yeah, are you saying my specimen seems large for those or small?
Yes.
Yeah,
Yep.
okay. Yeah. Yeah. Hmm.
Yeah, that was my thinking on what is it, Kretschme Maria? I can never remember if there's an R after the K, um, but the thing that's a little bit unusual, I think, is that these sort of nicely spaced little pimples on the fruit bodies. I'm not sure if that's a Kretschme Maria thing. But you can look that up.
Yeah, the only I don't think it's like a de Deleopsis type thing because it doesn't have the kind of eyeballs more or less. Um, there is one, I think it's annual hypoxylon, ther, the one that starts with TH, Therizinius or something like that. That one's a little larger than the other species, but they're more uniformly spherical, so that doesn't fit. But
That these like Jack Rogers Ella and annual hypoxylon. I think they all make these like small globs that are like a centimeter maybe or less, and these are kind of variable in size.
let me look that one up real quick.
When I see the Crusher Maria, it's always more in that light gray stage.
Right. That's where it's usually photographed because that's a more distinctive stage. When you see it in that stage, there's not a whole lot of other things that look like it. So one of them's the anamorph, one of them's a teleomorph. I forget which is which.
Okay. Well, yeah, it's kind of interesting those the pattern on those blobs. Okay, um, so we were looking at Pluteus, and they have with Dave, and they have the free gills, right?
Yeah, these are not. These are Entoloma. These
Okay,
gills are attached. They're
right.
Entoloma.
So that's what I was hoping, and um, I'm still just kind of trying to get beyond calling everything with that kind of pinkish look Pluteus, and the cap, sort of looks similar to like Cervinus or something. So Entoloma for sure. Okay.
Yeah,
That
if your hand is better and you're able to more easily do your microscopy, um, it's pretty easy to distinguish Pluteus from Entoloma by if you can get some spore drop, that's the best
for
because then
sure.
you have a nice clean look, without a lot of junk in the way. But even from a smash mount of gill material, you're going to be able to tell the difference in the spores. I mean, Pluteus spores are smooth and subglobose usually, and Entoloma spores are always angular. They're like
Exactly.
little polygons with a great big apiculus usually. So
So true. Yeah.
yeah,
And this I have to confess is just out of a file called Mushrooms to ID that I have it's from northern Wisconsin a while back. And so I'm just kind of trying to clean that up. So no opportunity to do that with the spores, but it's um an excellent reminder on that. And I will try. And my hand was doing a lot better, and it's not doing that well again, unfortunately, but hopefully it'll get better again and I can do more microscopy. But yeah, just thought I'd try to put some names on some stuff in my files. So, and then get clear on these pink gills. So, but the Antheloma gills aren't always this hue, and the Pluteus too won't always be this hue, right?
Well, the Pluteus gills are always rounded and come back on themselves, like before, prior to getting near the stipe.
Like obviously free, right, Dave?
They're obviously free, except when they're very young and everything's bunched together. Then it's a little hard to tell. And quite frankly, there are some Entoloma species, in particular Entoloma strictius group, where the gills can be very, very narrowly attached. Like some sources call it sinuate, and other sources use the word sinuate in a different sense, which is confusing. But in with the strictius group, sometimes it's just like a thread that really connects to the stipe. But another thing you'll notice is that even though the gills might taper down to a very, very narrow attachment, they're not rounded back on themselves, sort of. So it's not like the part of the gill closest to the stipe is round and goes back, that aspect, when you see what I mean yourself, you'll get to understand that.
Are you referring to like that's like they're adenate like coming in at 90 degrees? Is that
Well, no, but the Pluteus gills they're rounded. The end of the gill closest to the stipe is rounded, and so
okay?
the part that's closest to the stipe is part of this rounded part that is clearly not attached, and then underneath that the edge goes back out towards the margin a little bit. I mean, not close to the margin, but it just sort of bends back in that direction. There are differences in Pluteus gills and Entoloma gills that you'll come to understand just by making a lot of observations. And the microscope will help you there a lot, because then when you're not sure, you can just find a few spores and you'll know.
Right. Would you like venture to put in a species name on this Antheloma?
Nah, but I would say maybe you might want to start with like Griseus or something like that.
Okay.
But Entoloma is really, I mean, if you go on Michael Quebec, you'll see there's like 50 Entolomas or something like that. And that's certainly not all of them.
Okay. Thanks.
Okay. And then I found this yesterday.
Oh, is this one of those
Wow.
kiwi things?
Yeah.
Nice.
Oh, nice. Kiwi bubbles. Yeah, there it is. Oh, nice.
You can see these are about 1.5 millimeters in diameter. So they were on this stick. You can see they appear here starting at the bottom of the stick, and they go up pretty far on both sides.
Oh, so on a pretty thin stick. How thick is this stick?
It's about 4 millimeters thick here at the base.
Oh, wow. Okay.
And maybe 2.5 up here too. So you find this on really twigs and very, very small sticks. And this is a uh, large mature beech tree. And I had to look all around the base before I found this one stick that had the bubbles on it.
Well,
It's a standalone too. That's the thing. That's nice because all the beach that I see, they're in the woods with a bunch of hardwood debris, leaves, and sticks mixed in there. So it's cool you were able to isolate what that thing was.
Yeah.
yeah, and the one time I looked around for this thing, not very hard, but I was in woods with a lot of beech, and I was looking like at the trunks, you know.
Mhm.
So,
No, 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 no. It's only
ah,
on little twigs.
And do you think that twig just came down, or was laying there for a while?
It came down in the windstorm.
Okay, so this thing was probably on the canopy,
Yeah,
and just came down. Okay.
yeah.
Yeah, I've been looking right there at arm's reach and eyeline twigs, and I haven't been able to find any.
Well, if you look behind this tree, there's this row of beech here, and there are a couple here that they're fairly good size. And this beech here is a foot in diameter at chest height. So these, it's getting up there. And I found nothing on the twigs all through here. I went searching all through here and there was nothing.
Just on the one that was on the ground already.
Yeah, and that one was right over here. And I looked at probably 24 twigs all around the base of this tree, and that was the one that had it. So
And is this thing healthy living?
yeah, yeah.
Okay.
Now the other thing I found at the base of that tree, let's see. Let me go to my observations here. Was this guy. So let me back up a little bit. So this is,
Wow.
here's the base of that same beech tree. And if you look here carefully at the roots, you can see orange here along the edge, and there's some orange in through here. So if we go get a close-up of that, this is what those roots look like. And you can get a sense, so these are millimeters here, so this gives you a sense of scale.
Well, I think this one answers my last question. Is that tree healthy or not?
Well, it's certainly not on its last legs by any means, but yeah, it's got stuff growing on it, that's for sure. And I've seen this growing here for several years. So
What is that?
this is
It's one of the orange cankers.
Endothea gyrosa.
Thank you.
Mm-hmm.
That's neat.
Yeah.
So you didn't get a name change on that, right? Through INAT? Because I've had a couple name changes on things that I've called Endothea gyrosa, getting
I
new
think
names applied to them.
I think this is the current name. Uh, old observations on the INAT used to have Amphilogia gyrosa, and they got renamed as a group. But if you look at a map here, you can see this is fairly common throughout New Jersey. It's been found all throughout New Jersey. So
Endothea is a funny genus name. It almost sounds like Endophia.
yeah.
Endophew. Well, I don't know about this fungus, but my guess is that all the beech trees are going to be wiped out by the nematode before the fungus gets to them.
We'll
So
see.
enjoy seeing these critters while you can.
Yeah. I've seen this on, what else have I seen it on besides beech? The other one, the diatribe virescens, that only occurs on American beech, Fagus grandifolia. This one doesn't. This occurs on various species of, I think it's all broadleaf trees.
You're correct, Mike, because beech does not occur in the heart of the pine barrens. The soils are too sandy and acidic for it.
Uh-huh.
There is absolutely no beech trees in the pine barrens. None. Zilch.
So the fact there are little red dots here in the middle of the pine barrens tell you it grows on other things.
Probably oak or birch. That'll be my guess.
Uh-huh.
Or sassafras, or what is in there? There's like another tree. I forget its name. Has funny leaves. Tupelo. Yeah. So is this one of these erumpent
Yes.
fungi?
Yeah, and you can see that. Let me enlarge this for you here. So you can see it's coming up through the bark. So this is actually related to, it's a canker, so it's related to the chestnut blight.
Uh-huh.
Yeah, and I just checked a couple of my observations of things that
See here.
I'm now calling Endothea. But a taxon split happened as recently as 2 months ago.
Right.
Yeah.
But Mushroom Observer was ahead of INAT on this one. So
this is one of those, oh no, not again fungus, right?
yeah. So this one, these are younger stroma here. And here's what it looks like when it's older. You can see that it gets flattened and sort of felty-like and turns darker.
So are these classified as basidiomycetes or ascos, or what are these
These
things?
are ascos.
It's an asco. Okay.
Yeah. It's in Xylaria. No. I'm wrong. That's diatribe. What are these? I'm not sure. Let's see. Let's see if we can find out. So it's in the family. How do you pronounce that? Cryphonectriaceae.
Yeah, so Cryphonectria is one of the other splits from, well, they spent time in Chrysonectria.
So it's found on a variety of hosts, including, this is oak, beech, maple, what's liquid
Sweet gum.
amber?
Yes, that's a sweet gum.
Yeah, sweet gum, that's right.
And sweet gum and obviously maple are both present in the pine barrens. So
Yeah.
there you go.
Ilex is holly, Vitus is grape, and Prunus is cherry.
Mm-hmm.
And
Grape
grape,
is yeah, cool.
grape would be the vines
Yeah.
going up the tree, right?
Yeah, big woody vines.
Okay. Yeah, I think a lot of these things kind of all get called the same thing. You know,
So it's an
they
ASCO. Yeah.
anyway, like I was saying, these all kind of get called the same thing at the same time, whatever name is being applied at any given time. But as we
Uh-huh.
can see here, there's a bunch of things going on.
Right. Okay.
A quick question to for Jason: is sweet gum native to the Pine Barrens, or was kind of introduced there?
Um, it's common on the coastal plain. It's not particularly tolerant to fire, so it moves in. It's an early successional species that will move into a farm field after the farm is abandoned. So not introduced intentionally, but likely wasn't part of the Pine Barrens flora before we screwed up the fire regime.
Right. I noticed young sweet gum trees at a place where there was a lot of logging and also fire, and it seems to be
Yeah.
like on the periphery of like an open space.
Yeah, it's a pioneer species. It's more abundant around the edge of the Pine Barrens, but it's one of those early colonizers that will move into an area after disturbance.
Okay, thank you.
And on that note, I've noticed that there's a lot of ornamental sweet gums. Like they seem to be being planted in like parking lots and things like that. Um, is that good? Is that a good species to be planting in
I mean, it's
South
not
Jersey?
bad. I mean, it's native to New Jersey, so it's
Yeah.
not
Okay.
invasive, but as a parking lot tree, it creates those balls, which can be kind of a pain to clean up. So
Yeah,
it's
tell me
not the
about it.
tidiest
I've
tree.
been
Yeah,
doing
that's
this
why.
on the double here
That's why.
on my
That's
parking
why I
lot.
noticed them.
Yeah, yeah.
It's
The
interesting that as our climate got warmer here in New Jersey, these trees began to produce more balls. I mean,
yeah,
thousands.
the only worst place to plant a sweet gum in a parking lot is next to a pool.
Yeah, get ready for a lot of lawsuits and stuff. I think in some states or municipalities, this tree has been forbidden. It's been like been cut down and prohibited from being planted. So maybe these ornamental types are not producing the balls. I think you can control the proliferation of these things by injecting some hormones. Is that true, Jason?
Maybe I don't think that's economical. It might be true, but I don't think it'd be particularly practical. A lot of trees that are planted in parking lots don't live as long as they do in the wild. So, if you find a planted tree that's not producing fruit, it might not just be old enough, or it might just be, unhappy and not able to create the nutrients to create the balls.
Yeah,
Mhm.
because they also look wildly different, even from young ones that I see elsewhere. In the woods. Are they're all the same species? Is it what is it, Stylifera or Stellifera?
Yes, Terrassiflua. Yeah,
Oh,
there's probably a whole bunch of cultivars that are clones that are planted ornamentally. It's the reason it looks so different than the straight species.
So I'm wondering if there's anyone else who has observations they'd like to share.
I could share something if we run out
Sure.
of stuff to talk about. I don't have a specific observation, but I figured I could talk about the case of Usnia hirta in New Jersey. So if you search the genus Usnea in New Jersey, you get these 5 species on iNaturalist. The most common species is Usnea strigosa, which I agree with. And then the second most common species is Usnea hirta. But, if we look at the map, you can see it's pretty much statewide, not so much observed up in Sussex County. It's probably just because there's less Usnea, period. But all over the Pine Barrens and the Piedmont. But if you go to the 2019 revised checklist of lichenized fungi, New Jersey, by Dennis Waters and James Lundemer, which is based on herbarium specimens, and you go down to the checklist, it has 8 species of Usnea, none of which are Usnea hirta. And if you look at herbarium specimens, we've got 2 collections, one by Beadle, the other by Frank, from 1966 and 1986. But neither of these have been re-examined. This says the determiners, the collector, Emily Frank. I believe this one says the same thing. No determiner. So these haven't been re-examined by Dennis or James to confirm their identity. And if we look at the herbarium range map, you can see it's fairly widespread in Eastern North America, certainly more common further west, but certainly not as abundant as iNaturalist would have you suggest. So here's the iNaturalist distribution of Usnea hirta. So I think it probably is in New Jersey. I don't know of any record that confirms it's in New Jersey, but I think this is a case where early on in the iNaturalist life, somebody uploaded a few observations of Usnea and misidentified it as hirta, and then the algorithm, the AI in iNaturalist, kind of picked up on that. And now if you observe an Usnea species anywhere in the state, Usnea hirta is one of the top recommendations, and it's kind of this snowball effect. So it's kind of an interesting case study on the dangers of iNaturalist and AI, and the accuracy of iNaturalist identification and how dependent it is on the data that goes into the formula. I do know of a high quality record. This is by John Franklin. This is up in upstate New York. You can see the species is Isidiate. But somebody's got to go through all of the Usnea hirta observations from New Jersey and either downgrade it to genus level or looked for a definitive Usnea hirta observation, because I've gone through a few of them and I haven't found any yet.
So do you think a lot of the ones that we get in New Jersey are strigosa without the apothecia?
So I think these 2 species need to at least get switched. I think Mutabilis is the second most common species in New Jersey. And the best way to tell those apart, obviously Strigosa often has apothecia. Mutabilis almost never does, if ever. And then if you stretch out one of these stems and look at the medulla or the medulla, in Strigosa, it's white, and in Mutabilis, it's reddish pink. So if you don't look on the inside, and at least get a color of the medulla, then you really can't put an ID on these things unless it has apothecia. If it doesn't have apothecia, you have to look at the color of the medulla.
Can you enlarge the mutabolus?
You can see here it's got more Ceridia than it does, Isidia.
Got you. Yeah, I heard you say it's isidiate. Is Strigosa isidiate, or is it also just kind of dusty like that, ceridiate?
Let's see. So it's definitely bristly. Whether or not you would call these Isidia or not, I'm not sure. It's not impossible, but unusual for a species to be both apotheciate and isidiate. Let's see. We go.
So only some lichens have apothecia?
Correct. Yeah. Some lichens reproduce sexually, some reproduce asexually, and a few reproduce both. Let's see. Let's see where it says apothecia.
Is Jason's talk on lichens that he gave us, is that up on YouTube now? Is that
On calicioids, it is.
No, not the recent one. The one you did like last year. Do you remember? It was not lichens. You talked about
I don't know. I'll have to check. I don't remember that presentation.
Okay. I thought it was you. Maybe it was not. I might be confused.
No, it could have been me.
No, it was Jason. I think you went over all the lichens, like the groups of lichens. It was a very in-depth, presentation. Very nice.
It was. Yeah, I'd like to review that if it's out. So I don't know.
Well, I think Lila said recently that she finally got control of the YouTube channel from Luke, so I think all these recordings are available.
A lot of them are, yes.
So they're probably archived. And
Yeah,
it's
so
just,
we're in the process of uploading videos.
Yeah, there's a few presentations that I did that for one reason or another I asked not to have published on the YouTube page, but it's on YouTube and available to share if somebody requests it. So that might be one of them. If it is, I don't remember why I would have not wanted it published, but
I understand that.
If it is up there.
Okay. I'd love to see it again.
So, Christopher, you said you had something to share?
Yeah, before Chris gets into it, thanks for sharing that, Jason.
Yeah.
I'm pretty sure I have something up on iNat Hertus, so I'll address that.
Cool.
Yeah, I was just looking through a number of New Jersey observations that people have attached the Herta ID to, and they did not look like Herta to me.
Well, since we're here.
Ah.
Wow.
There was dozens of these that had blown out of the trees all over the place where I was.
Where is this?
A lair.
Yeah. Yes, this is definitely Usnea strigosa because it has that apothecia.
That is so cool. As soon as you started talking about it, I was like, oh, I forgot to put that thing up. So I just put it in.
Wait, can you zoom on an apothecia for me? Oh, yeah, duh, right there. I thought there was a glare on my screen. Yeah.
Strigosa, huh? Awesome. And then I have another interesting photo.
Ah,
This was also at a lair. I cannot believe how long these persist. This is like I saw like 3 of them.
What is that?
I forget the name of them, but I think they're one of those ones that might grow on another thing.
You think it's a telepicladium?
Oh,
Yeah, maybe.
Hmm.
is
I,
it pretty small, pretty tiny?
I think that's a capitate stipitate mushroom, maybe. Immature one.
I don't remember what it was. I just snapped a photo of it. I was gonna dig at it, but it turns out that in that hard ground like that, what you do is you just slice it right off at ground level, and no digging in frozen ground.
Yeah, it hurt. Yeah, right.
thank you, Christopher.
Yeah, thank you.
Does anybody know the genus Cyclocybe? I just
Heard of
I,
it.
Googled
I know of it. Yeah.
Yeah.
And I've compared many things to it.
yeah, I Googled like mushroom with black cap for the one that Chris just shared. This one looks like it's a maybe. There's some observations from the winter.
I thought it was one of those snow-loving bullets, but no such luck.
Yeah, but do you guys agree that looks like a capitate, like a actual mushroom and not a telepicladium type thing? Not an
Yeah, I mean,
asco, but a basidio.
it would be nice to see underneath the cap, but the overall, silhouette of that thing was, yeah, pileate stipitate mushroom.
Yeah, it could have been like a stropharia, like a wine cap or something, or maybe a Cordonarius violaceus or something. How thin was the stipe, Christopher? Like how
Like a good almost 2 centimeters wide. It was like that chunky little thing, and the cap just kind of on those just kind of like meets the stem.
So thicker than a pencil, would you say? Like double a pencil?
Like double.
Yeah, because the deer truffle ones, the hypocladium or whatever. I mean, the ones I've found they're not any thicker than a pencil. The stipes. I don't know if other people have found larger, but
Yeah, did you say, Chris? Did you say like 2 centimeters that stipe was, or the cap?
Yeah, probably.
Yeah, maybe a little bit bigger than a pencil hunter.
More like 4 pencils.
Yeah, yeah. But right, no bigger than 2 pencils. I'll say.
I mean, 2 centimeters in width, that's like almost an inch.
Yeah.
yeah. So that's sounds like a real mushroom to me too. That's cool.
Yeah, it's kind of difficult to imagine a pileate stipitate fungus growing in the middle of a winter, especially a harsh winter like that. I've checked all the weather almanacs in a website that I pay for, and an analog year would be 2003. It's been that cold consistently throughout the entire winter. So
Or like in the 2 pencil range.
now it's a 2 pencil range. So it's a centimeter, eh?
Listen, I use a lot of inches in my work. Like 2 pencils, not it wasn't super thin, but not like a bully.
What was it? What was under the snow there? Is that wood chips or
In
what
that
was
spot,
it?
just sand.
Oh, hmm, interesting.
So why didn't you collect it or cut it or whatever?
Oh, because when I went to collect it, like I said, I sliced it right off at the base because I couldn't dig into the ground, and it flew right into the woods.
Yeah, but did it have gills? I mean, it's kind of strange that you just snapped one picture of it.
No, it was gone. That's what I'm trying to say. Like it flung right away. Like the ground was frozen. So I went to dig at it, and it just sliced right off. Off it went.
Into brush and you couldn't find it,
Oh, I
basically.
was gone.
Yeah.
So as you was trying to cut it at the base, it became a projectile. It just flew away,
Yeah.
and you never found it.
Didn't even try.
Funny.
It went going in there.
Wow. Well, that's unfortunate.
Yeah, but I see ones that look like this in the fall in the same area. I mean, it's probably just been there.
So you're saying this is not like a recent fruiting. It just it's something that survived, for months, just sticking out of the
Yeah,
ground like that.
just persisting.
Hmm. Well, I mean, like I said, it's very difficult to imagine something mycorrhizal or saprobic that's pileate stipite that will be growing in the middle of this harsh winter with all the snow and nights in the teens.
No, I don't think it was growing at all.
It was frozen in time. Okay.
You guys want to look at Jason if you're still here? If anyone's still interested in looking at some usneas that we could talk about, I could share a couple.
Sure.
That's
Yep,
news
still
to
here.
you.
Definitely. And I did send Mike one other one, but after the Uznias, if he can share it.
I'll
All
check
right.
my email.
Can you guys see my screen?
Yes.
Yeah.
Yes.
Cool. Yeah, so this is one of the ones I would call this ceridiate, right? Although it is kind of strigose, it has those, jutting out tendrils. So, Jason, with the lack of apothecia here, do you think you can call that the one that starts with the M? I forget what the name of that was. I wrote it down.
Mutabolist.
Mutabilis?
I mean that. Probably 80% certainty. Yeah. I'd still want to see the medulla for sure, but
Okay, cool. But if I'm looking at something like this, I can do that medulla test and get an answer from it. Cool.
yeah, and you just kind of you can just pinch one of the branches with your fingernail and just kind of tug on it. They're usually kind of stretchy, and you can usually just kind of pull off the outer cortex.
Yeah, that has nothing to do, I think, with the actual lichen here. I think that's a insect sac of some sort, right?
Behind the hairy like structures, it looks like apothecia, but those aren't.
So, yeah, and correct me if I'm wrong here. That is squamules or the thallus of a different lichen, right?
Thallus, yeah, thallus of a different lichen. That's a folio species. Yeah.
Okay.
Something like maybe Parmotrema or something like that.
Yeah, or flower parmelia.
Okay.
Neat.
So zoom in on that picture again, and go down and to the right a little bit. Keep going. All right, there. Let's see. Where's your cursor? There, right there.
Looking at this.
Nope, go down. The other one. Go to the right. The vertical, so parallel to the one you were
Oh,
at
right
before,
here.
right there.
Right here. Yeah.
Yeah,
Oh, yeah,
that's a
yeah.
break in the cortex.
Okay.
That's a white medulla right there.
Cool. White medulla makes it
Not mutabolous.
strigosa.
Yeah, or something else.
Oh. All right.
There-
The not potentially the rare one or no?
Uh, yeah, I don't know. I mean, I'd want to get a better look at the medulla, but that's basically what you're looking at there. I mean, that looks like a break in the cortex. You know, I'd still want to do a cross section, just make sure that it's white all the way through. But you know, that's kind of what you're looking for.
Cool.
That is cool.
All right. Let's look at this one. I wanted to look at. So I was pretty sure this was strigosa, um, but without, I guess the medulla and perithecia, apothecia. Can't really call it, I guess, huh?
Yeah, I mean, it looks more like strigosa than mutabolous, but you know, these things are variable and difficult. You know, usnias give people a lot of trouble. You know,
Yeah,
you
because you
think
can see.
that because they're so easy to spot, people tend to think that they're, you know, a genus that they can focus on when they're just learning lichens, and they're fooled.
And then they start uploading things with wrong names, and
Yeah,
that messes up the algorithm even more.
yeah,
But you can see at least this is less ceridiate than the
right.
last thing that I showed, right?
Yeah.
All right, cool. I guess
The
those are
other lichen there has these cool like black
highlights.
hair-like structures. What are those?
So the black
Mm,
structures are called cilia, and that species is something in the genus Parmotrema. Those black cilia are the giveaway for that genus. Could be Parmotrema hypotropum. That's a common one that has black cilia, but there's other species that have it too.
That's pretty neat.
so
Actually,
Parmotrema.
if I can share my screen real quick. I
Yeah.
don't know if I've shared this before. Let me see if I can find it. On the topic of Parmotremas. And cilia. Um, Here we go. So these are the species of Parmotrema that we have in New Jersey. There's 12 species that have been recorded from the state, and then 2 bolo species. Be on the lookout for. These are things that occur in neighboring states and might be in New Jersey. For the most part, they all grow on bark. But here's a little chart that shows the different chemical tests for Parmatrema. You can see the diversity of tests, particularly P.
Can you,
on the
Jason?
medulla.
Can you zoom in or? Yeah, thanks.
So a K test on the cortex isn't going to tell you much, but a K test on the medulla or a P test on the medulla is going to narrow things down for you quite a bit. And then beyond that, you've got reproduction. Does it have apothecia, isidia, or ceridia? That'll help you narrow things down. And then cilia, absent. Those black hair-like structures on the edge is absent, abundant. Sometimes it doesn't help because it says usually present, not definitive.
And did you put this together from your observations from New Jersey, or is this a collection from other works?
No, this is the checklist plus information that I've gotten from literature on species descriptions. I've tried to fill in some of the holes with looking at iNaturalist observation and high quality IDs, but as you can see, I'm still filling it out. But
it's really good work, though.
But yeah, so since we're talking about the cilia, so hypertropum abundant, as opposed to Gardneri, where the cilia are very sparse.
And can you talk about what's the spacing look like with abundant and very sparse versus sparse? Is that pretty self-explanatory, or is it,
Well, let's look at an example of one that has sparse cilia.
Yeah, hardly. You got to look for one.
Yeah, I'm not sure I see any. You check Jason Hollinger's. He's got pretty good pictures. Parmatrema doesn't have. There's Gardner. Let's see. Yeah. So yeah, that's kind of my way of trying to wrap my head around these things, because this is another example of where people throw out the name Parmatrema hypertropum probably way more than they should. Although in this case, it is probably the most common species in New Jersey, but it's guaranteed to being supplied on iNaturalist to species that is certainly misidentified.
So those T's are silent in those descriptive words at the end there. That's good. Good to know.
What T's? What do you mean?
The T's in those characteristics. If you bring your sheet up again, I can say it, but like Ceridia.
Yeah, so ceridiate is the adjective, ceridiate species, and then the name of the structure is a ceridia.
Got it. Okay, thank
The
you.
species that has ceridia is ceridiate.
Thank you for that.
Yeah.
Is that a plural or a singular?
Plural, ceridia.
Mm-hmm. Okay.
Ceridium, singular.
Yeah, I guess, but I don't know if you'd ever have a reason to, if there's one ceridium, there's going to be a
Well,
billion of them.
some of those cilia, I was looking for a cilium.
Yeah, not a single cilium.
Yeah, and so the one that Brandon had behind his the lichen, he was really showing that that looked like abundant cilia.
Yeah.
Yeah, quite abundant. So with lichens, they have often it's ascomycetes that are involved with them. But do we not often see those that structure at all? Because they're so tiny or something?
The what structure?
Like a fungal structure, ascomycete. Because aren't like lichens often a combination of bacteria and fungi and algae, but like ascomycetes are the fungi, right?
Yeah, so lichens will reproduce either sexually or asexually. So if it's an asexually reproducing lichen that has Ceridia or Isidia, then it's not reproducing sexually, so it's not creating spores. The spores would have to come out of an apothecia. So if you take a cross section of an apothecia, which sometimes you do, and you mount it, you can find the ascospores, and you have to find, is it, 4 or 8. Um, but
Okay.
that those are in those discs that are created. Sometimes you got to take a very thin slice of that and put it under the microscope to count the ascospores.
Oh, very, very interesting. And so, because what's confused me are the cup-shaped structures. I've thought, oh, those are the ascomycetes, but they're not at all the ascomycetes.
Yeah, I
Okay.
mean the whole thing is an ascomycete. So yeah, the apothecia is the reproductive structure of the fungus. Um, but it doesn't always reproduce sexually.
Okay. It's a little bit confusing for me, but really interesting.
So what he's saying is you can't separate the fungus from the lichen. The lichen is the fungus is the structure of the lichen, and inside that greenhouse is the algae or the cyanobacteria, right, Jason?
Yeah, so by weight, probably 90% of a lichen is the fungus. It's just that thin layer of algae in the middle. The you say the lichen's a combination of a fungus and an algae. If you take a cross section of a lichen, you can see the algae layer. They're not mixed. They're compartmented.
Okay, and then those disc-like structures are apothecia, but sometimes there's like in the green, bright green one that I showed, it looked like these like tiny little cups. Are those also apothecia
Yeah,
or
I think so.
okay, okay, thank you.
Yeah, you can get apothecia of different shapes.
It's an interesting conversation because, even, when we have these conversations, or with lichenologists, Dennis Waters, Jason, we were there at that presentation that with the Philadelphia Botanical Club. That relationship really isn't that well understood. You know what I mean?
Right.
it's described as this symbiosis, but it's a fungus kind of controlling other things, in my opinion. But
Yeah.
and spreading fungal spores. It's mostly it behaves like a fungus, in my opinion, lichen do.
So, share my screen one last time. I just put a hunter's yellow lichen on the iNaturalist page, and this is the name that came up: Pleopsidium flavum.
Thanks.
Which, let's see, look at the map.
Yeah,
Yeah,
what's
that
that
makes sense
distribution
for-
look like?
Yeah. In the West.
Yep, Western species.
Can you click on one of the ones from the east, our area, maybe, to see if maybe it's a misidentified thing?
Oh yeah, that's
Yeah.
rhizocarpon with black apothecia, so it's definitely not right.
That's a riot.
That's
That's not it, right?
not it. Don't know what it is, but not it. So, yeah. Whatever that is leprous. Dusty.
Wow.
Yeah, so that kind of goes to show you how even that distribution map, you got to click on these things, right?
Yep. Especially with
Really?
lichens. Even some of these in the Midwest. Yeah, I mean, look at that.
Is that a research grade one too?
Yep.
Yeah.
Hmm. But you like that name for my Arizona one.
Yeah, I think that's probably what it is.
Okay, cool.
Here, all right, let me share my screen one last time.
Sure.
So here, this is a website, Italic 8.0. This is the lichens of Italy. And I found that over half of the lichens we have in New Jersey are profiled on this website.
Wow.
So it's definitely worth bookmarking. So here it is for Pleopsidium, where it calls the thallus crustos slash plycodioid, bright yellow, somehow glossy, central areoles, varicose, and then apothecia half immersed in the thallus, the brownish yellow concave flat disc. So you've got information on chemistry, notes on habitat. So it's a nice, if you're trying to understand what you're looking at, are those things apothecia? Is this thing a crustose or a folio species? You can kind of look it up and read the description and see how they're describing it. And then they even go into distribution maps on lichens of Italy and predictive model of where it would occur, along with photos. So this is a pretty well done website for a pretty large number of species.
How well, do you know how many species they have of lichen in Italy?
No, let's see.
Because in like New Jersey, we have what, 700, 500?
Right now, the list is up to 533. Oh, right. They also have these identification keys. I haven't used these too much. If you go to, the key of Peltigira, it's like a photogenic, that was dark, that was bright green, dark. Ceridia over here, Isidia over here.
That's cool.
So it's like a picture key. One day we'll have this for New Jersey.
So it's interesting that a lichen would appear in Italy and the Western North America, but not Eastern North America.
Yeah, I don't know. I mean, what is it? The Alps in the northern Italy might be more congruent with the Rockies. I don't know. I'm not familiar enough with.
Yeah, whereas more northern Europe, Scotland, Ireland might be more northeastern U.S.
Yeah, I mean, I think a lot of lichens are common and they're not particularly tied to a specific habitat. So, um, and then a lot of the species that we have that don't occur in Italy are species that were described from North America. So, how much of that distribution is taxonomic versus ecological?
Yeah, that looks like a really great resource. Besides that website for lichens, do you recommend anything else for lichens, web wise?
The
Yeah,
consortium website is really good, right?
yeah. So before I answer that question, I just looked up how many species of lichen Italy has, and according to the AI response on Google, it says 3,000. So they got about 6 times as many species as we have in New Jersey.
Wow.
The I should really add more websites to the NJMA's lichen webpage. There are some other resources that are worth checking, but yeah, the consortium and Italic are probably the best in terms of looking for descriptions, and the consortium's herbarium records for distribution, and then ways of enlightenment for photos.
Cute name.
Yeah.
All right, so I got to ask because Italy has 3,000 species, right? That's crazy.
Mm-hmm.
It's not a huge country. Do you think that has anything to do with the age of the country? And, Europeans were the first that started classifying fungi and ascomycetes and lichen and things like that? Or do they really have that much more diversity than we have here?
I think it's probably both. I mean, Europeans are miles ahead of us on bryophytes and lichens. But also you're going to have, um, let's see, what, yeah, so Italy, ranges from sea level to 15,000 feet. So I think that elevational diversity is going to be a big contributor.
Cool.
New Jersey only goes up to 1,800 versus 15,000. So Plus, they've got islands in the Mediterranean, so there's probably a lot of diversity there.
Yeah.
I wonder too if the architecture lends to more like availability of specimens, just because it seems like their structures are still stone, like so many of their structures are still like this ancient stone buildings that the lichens would hang out on, where we don't have that as much.
Yeah, maybe, but the problem with historic structures as substrates for lichens is that you can't easily collect those species to study them without chipping off a piece of the historic structure, which you typically can't do. So those historic structures are likely undersurveyed relative to natural areas.
Okay. Well, it's really fun to learn about them.
Well, it sounds to me like we've come to a good stopping place for this evening. Unless anybody has a last thing they
Mike,
want to
did you-
share
I
or a
do.
question they want to ask.
I
Well,
actually sent you one thing
Okay.
to Taxonomy Tuesday. It's an observation from Mushroom Observer.
All right.
and I sent you one thing too,
Oh, that's right, Hunter.
but Igor should go first.
I'll be quick. It's just like a small note.
Okay, just a minute.
And before we get to that, while you're doing that, Mike, is every Fissia in New Jersey Fissia Malagrana?
Oh no,
No. Okay.
but milligram is probably the most common.
Okay.
All right, let me share my screen.
Okay, so this modest-looking small mushroom was collected by Nina Burkhardt in the Pine Barrens at FPP, the Franklin Parker Preserve. Well, almost like wintertime, late March of 2023. And I mean, you can see the pictures here. I mean, they're okay, but it's a small mushroom, probably not in very good shape. But you can get the idea. It's, somebody would say just an LBM of some kind. Not easy to put to genus. It has attached gills. So I think Dave proposed Tribulurius for this thing, and it was fairly quickly forgotten, except that it was submitted as one of the lucky ones for sequencing to Stephen Russell, one of the 168 collections. So this mushroom was sequenced, and the sequence failed. The sequencing failed, and now Steve is going over all the failures, not just ours, but everything that he failed to or overlooked to sequence. And this mushroom yielded a sequence, a full-length ITS sequence, quite good. And the sequence is brand new. It's brand new to GenBank. It's brand new to Stevens database, and apparently this thing is a cotoscopy. So if you imagine your cotoscopy something with the current gills and looking more robust and different, maybe sort of like ampular cotoscopy. But cotoscopy is variable. But I would not call this thing cotoscopy. I mean, some obscure genus. Who knows what this thing is? But the DNA says it's cotoscopy, and I looked at the blast, end results, and there's tons of cotoscopies in there. Some good identifications apparently.
Igor, thank you for sharing this. Dave emailed me this observation earlier today. I shared a thing recently in a very similar habitat that had very similar microscopy, subglobospores. I also couldn't get a spore print out of it in a burned, conifer pine bearing setting.
Yep.
Yeah.
Came from a
Cool.
burn area, as the notes here say. You can see in the notes section. Yeah, kind of this, little obscure, easy to miss, unassuming kind of thing. So yeah, interesting result. What do we know about mushrooms, right? So a new sequence, a new species, probably. I doubt this was described. It's just too easy to overlook. And maybe it's just found in the pine barrens, in a burn area.
In a burn area, yeah. And only shows itself, after that burn, X amount of years later. So yeah.
Exactly, and maybe it doesn't like to grow in a warm time of the year. Maybe it's just likes cool weather, so you only find it like in the fall and spring.
And then also you can combine that with we had a very dry season. So does this thing love the winter after a dry season in a burn, and no one looks at it because it's the middle of winter? Cool.
Yeah,
this was from 2023, right?
Yeah, this is from March of 2023. 1 centimeter cap, 2 centimeters stipe. So given its colors and blending in with the environment, you probably easily overlooked.
And then I take that back. 23 was a decent season.
No, 22 was actually a dry summer. The fall was not as dry. I think it was average, but the whole season was basically damaged by the drought of 2022. And so, yeah, it sort of came out, on the heels of a major, rain deficit in summertime, although it was somewhat compensated in the subsequent months. So that's it for me. Clitosopy, species New Jersey 01. The first one of its kind.
Amazing.
That's awesome.
Clitosopy is one of those things. I mean, if it doesn't, that's crazy that it's a clitosopy. Look at that
crazy,
gill
yeah.
attachment. Look at the gill attachment, right?
Yeah, exactly. It has probably a notch as far as I can see. Deep down, and it's not even decurrent or even subdecurrent.
No.
Yeah, I would call it just some kind of a cyclosopy or croposopy or whatever. So, cyclosopy. Yeah, that's a genus where everything like that's unknown goes. Like something obscure, crummy looking, and brown and LBM. Yeah, MBM like.
Or formerly clitosopy, right?
Yeah, whatever. Exactly.
So this is why it pays to go out every month.
Yeah, and also pays to sequence these boring-looking mushrooms.
Right.
Because chances are, if it's boring and small, it's something that's completely unknown.
Right.
Can I ask a question on the measurements for the stipe on that? Can we just look at those again really quick? It said like what does it say?
Millimeters.
2 centimeters by 1 millimeter. Like how am I reading that? It's only 2 centimeters tall, or?
Yes.
Correct. Okay.
So this is a very tiny mushroom.
Very tiny. Yeah.
I'm not sure about 1 millimeter, but the pine needles are certainly wider than 1 millimeter. So Mina probably just mistyped something here, but the stipe is probably a few millimeters thick. I would think maybe 4. But
And
the
only
stipe is
2
longer. I don't
centimeters
know.
tall, though.
How many pencils is it?
I know.
How many pencils? I think it's a half a pencil thick, maybe a third of a pencil thick.
Okay.
Yeah, cool.
Thank you. But it's like really short. I mean, is it truly only like how tall is it?
Yeah, well, if the cap was measured correctly, 1 centimeter, I would think that the stipe is probably around 3 centimeters, no more than that. And the width of the stipe would be, I don't know, maybe three or four. Maybe, yeah. I mean, it's look at the thickness of the needles, so that's your reference point. Needles are, I would say, about maybe 2 millimeters thick.
Yeah, those are like white pine needles there, or no?
No, these are pitch pines.
Pitch pine. Yeah.
Okay.
And also, I think the question on this is what's it growing from? Because I don't think we got the base of this thing.
Well, there was a discussion. If you scroll down, there's going to be a discussion between, I think Dave.
Yeah, is it a Strobelorius, right?
Yeah, he was asking Nina if it was buried or growing in a pine cone. And I think Nina never replied. Maybe she never got that email because we had issues on the MO with the communications. Emails not getting through because they were never actually sent out by MO itself, as we recently found out. So yeah, there's Nina maybe doesn't say anything about that in the comments section. I don't know.
But I mean,
So probably growing from the ground.
Yeah, I would say it's probably growing from the sand. That would be my guess. It says does not appear to be on wood.
That's super exciting.
Right. Yeah, the closest hidden gem bank was a collection from Europe called Taxonomy Phoetons. I forget which country, Italy or Austria, something like that. Yeah, you can click on that link. It's going to bring out the gem bank accession, and you can see what country it came from.
And Igor,
down.
I think I saw you say that match was what,
France.
95 to
96%. Yeah, 96%.
Okay.
Yeah, so this came from France, and it was published actually. So that species name could be correct for that accession. That is for the French accession. Phoetons. So
cool.
Anyway, so little things. Yeah.
Yeah, so I showed this last time, I think, and I don't think we had. I think we looked at this Mushroom Observer entry that was a joke, and they called it
was
like
a joke. Yeah.
Globus
Globus albus. Yeah.
Yeah. Well, I was.
It's Helicogloia, right?
Oh, well, I was thinking it was Spherobolus. And based on, I was doing the quizzes on Michael Quebec, and they had a picture that looked just like this, and it was like the young Spherobolus before it shot out its stuff. But what did you say you thought it might be? Or did we decide it was what you said, Brendan?
I mean, it looks like Helicogloia compressa.
Is that a slime mold?
No.
Okay. Thank you. I'll look that up.
Did you find this?
Yes.
Did you touch it?
No.
No.
I did not.
Yeah.
Thanks for spelling it, Jason.
I mean, looks pretty good for Helicogloia compressa.
He put it in the chat, Hunter.
Yes, I saw that. Thank you so much for that. Okay, good. Well, that's great. I thought I could do better than the Globus, whatever. And so I just wanted to show it again. So that, and that I'll look this up, and if it's not the compressor, maybe it could be a young Spirabolus, but I'm looking forward to checking this out. I think it's probably more likely. So thanks.
Wait, what did you think it was? How do you spell that? Strolobulus?
Is Spirabolus? It's like Sphaerobolis, I believe.
Sphaerobolus, gotcha.
I mean, it's usually not shown looking like that, but then this picture on Michael Quebec, it looked a lot like that. I took a screenshot, but it's not on this device, but I mean, it really looked like what I had. Did that spelling work for you? Did you look at it?
Yeah, the cannonball fungi.
Yeah, that's right. Yeah, which looks nothing like it, but except in a few pictures, it sort of does, like when it's really young. Do you see any like that or not really?
Somewhat, but it looks more like an immature Helicogloia.
Cool.
You should look that one up.
I'm gonna look it up.
When they get, well, actually, I guess this would be a more mature one. When they're more immature, they have more of a kind of glossy coating to them. But I think that's what you got there.
Sweet. Okay, good. I'm gonna look this up. Thank you. Oh, yes. Yeah, that seems like for sure. Yeah. Thanks. That's excellent. Appreciate that.
Yeah, that's one. Once you see it a couple times and you learn it, and you're able to see it through its progression from young to old, because it does start out as just kind of a gelatinous layer on a log, and then those gelatinous layers kind of grow into eyeballs, and then they turn into slimy teeth, which is not a not a scientific description, but that's what it is.
Okay.
Yeah, almost always well decayed logs. It likes the moisture, I think.
Thank you. That's great. I will make that correction in my files here. So, and then how does that release its spores? Like what kind of fungus is that?
Great question. I would assume it's an ASCO, but I actually don't know.
It's like a Basidio carp.
It's Basidio.
Wikipedia.
Oh, okay. Interesting. So yeah, I'm just trying to figure out where to put it in my folders.
But if you touch those things, so that's how you really learn them. They are super hard
Really?
and they won't wipe away. You
So
know what I mean?
here's the taxonomy on it.
And they just got a funny name. I saw somebody put a name to them on iNet, somebody had to come up with a name, put a funny name on them.
A funny name.
Where's the taxonomy? Are you sharing?
Yeah, it's right here.
A more funny name than Globus, bulbus, or whatever.
Yeah. I think,
Globus albus, white blob.
That's
hold
it.
on. Hold on. No, common name. Like someone had
Common
to
name,
assign
white blob. Yeah.
a common name to it. Hold on, I'll find it. Mike, are you going into the taxonomy on it, or
It's oh, sorry. I'm not sharing my screen. There, you should see it now.
Okay.
So in the Fungi of Europe book, they put it in the section of the book that they describe as pin-shaped to resupinate jelly fungi with transversely septate basidia.
yeah, I was going to say that.
You're right on it, Jason. And it's
Oh,
interesting. This is often found in fall and winter.
yeah, it was so the name that was applied to it was tiny hardballs.
Tiny hardballs.
That's funny.
And someone was forced to come up with that like a month ago, which is silly.
Okay, so I'm going to sign off. Do you want me to make somebody else the host?
Yeah, yes, you could.